agenda and the study update. So you can do second slide, Sohana. So the overview of the REVIVE trial, of which we are all part of, is going to be done by Dr. Thomas Shire, and he is at the REVIVE project office based here in Hamilton, in Canada. And then today's talk on sample size considerations is being done by Professor Shrikant Bangdiwala. He's a director of statistics at PHRI here in Hamilton, which we had the privilege of meeting recently. And after that, it'll be followed with a question and answer session in the end. You can also post any questions you have throughout the talk in the chat, and we will read it off the chat box in the end. Professor Bangdiwala has also been so kind as to share his presentation today. And would you want a copy of this, you can just pop your email address in the chat box, and I will share it with you afterwards. So without further ado, I will hand you to Dr. Shire.
So thank you, Elsa. I'm sorry for my bad video quality. It looks like I'm the only one at PHRI without any light in the room, which actually is not true. But it's actually an honor and an absolute delight to present you an overview of the REVIVE trial and also the progress we made so far. So on this slide, you see the pragmatic trial design of the trial. And if we go through the figure from left to right, we see we include adults 18 years or older with a low CD4 count and randomize them in a one-to-one way to either placebo or a 4-week treatment of azithromycin. And then we assess the primary outcome, all-cause mortality at 24 weeks. And the hypothesis, as stated at the hour below the figure, is that among patients with a CD4 count of less than 100, this 4-week treatment will reduce mortality compared with placebo at 24 weeks. And if you go to the next slide, we see how far we got so during the way. You see, like in the table on the left side, there's like a total of 14 countries where the REVIVE trial aims to activate at least one site. The number of sites is different for each of the country and sums up to a total of 60. In the map on the right, we see the geographical distribution of the countries where we are currently recruiting. These are the green countries, and where we are actually close to start the first site, which are kind of these yellowish countries. And for these yellowish countries, for most of them, it's just like a matter of a few weeks until they are ready to start recruitment, and we are just awaiting final approvals or import permits. And now I think the most important and interesting thing is represented in this table, which is the weekly recruitment report. And I think here's now also the time to thank all the investigators, co-investigators, and all the hardworking staff at the site, which actually kind of created these numbers we are now presenting in this table. So each row is like one country, and the green row at the bottom is like the total of the table. And as you see to the left, all the recruiting countries sum up to a total of 44 activated sites. And towards the middle of the table, you see the recruitment either like up to the 2nd March, and then like in each of these columns, which are labeled with one of the dates from 3rd March to 24 March, it's the recruitment for the corresponding week. And the most interesting numbers, I think, are like 2,057, which is the total number we recruited so far up to today, and 294, which is like the total amount of participants recruited in the last 30 days, which is like an absolute astonishing number. If you just like, it's like roughly like 10 patients a day get randomized into the REVIVE trial, pushing us closer and closer to the needed sample size of 8,000. And I think actually that's, if you go then to the next slide, that's also kind of then the handover to the talk today. If we see at this graph and the x-axis, there's like the time from when you started in May 2023 until the kind of the designated end in December 27. And on the left y-axis is like the total number of participants, and on the right axis is like the cumulative percentage of activated sites, which is highlighted in this greenish color at the background. But if we focus on the blue line, which is the projected cumulative enrollment, we need to have to achieve our sample size target of 8,000 patients by December 2027. We see that the violet, or like the purple line, which is the actual cumulative enrollment, actually is like closing up to the blue line, which means we are gaining ground to get back to the speed we need to have and the recruitment we need to reach our sample size. And so far, it's like, as you'll see here, it's a long way still to go, but we are kind of optimistic with this turn of the purple line, and we hope that we see an even better recruitment at the next seminar. And I think with this, I hand back over to you, Elsa.
Thank you very much, Thomas. It is always a pleasure to see, and delightful to see how this study progresses from week to week, and we the sites are really doing their utmost best and picking up speed at a great rate. So without further ado, I will now be introducing our presenter for today. And we had the privilege of meeting him in person, but it is an honor to introduce our distinguished speaker, Professor Shrikant Bangdiwala. He's a globally renowned statistician with over 45 years of experience in designing, monitoring, and analyzing multi-center randomized clinical trials. Professor Bangdiwala is a director of statistics at PHRI here in Hamilton and a professor at McMaster University in Canada, where he plays a pivotal role in advancing health research methodologies. Previously, he was a professor at the University of North Carolina, the Gillings School of Global Public Health, where he has been honored as a Professor Emeritus. Beyond his groundbreaking research, Professor Bangdiwala is widely recognized for his ability to make complex statistical concepts accessible and engaging. With over 250 research methodology courses taught worldwide, he has empowered countless professionals to apply sound statistical principles in their work. His excellence in global public health has been recognized with the American Public Health Association's Distinguished International Career Award. We are privileged to have him with us today to share his expertise on this critical topic. Please join me in welcoming Professor Bangdiwala.
Okay, good morning, everybody. Good afternoon in the African continent. And thanks for joining this seminar or webinar. It's a very appropriate topic as we just have heard that recruitment is going fantastically in the REVIVE trial. And so they are approaching the sample size quicker than expected, I guess, of 8,000. So the question that we're going to be dealing with is what are the sample size considerations for RCTs? And specifically, for example, why do we need 8,000? Where did that number come from? How did we think about we needed that large number? So, are you seeing my slides? Yeah, so hope so. So why we worry about sample size in general? The reason we worry about sample size is that at the end of any study that we do, we want to have adequate statistical power to test a primary hypothesis, okay? And remember that an RCT, a randomized controlled trial, is designed to answer only one question. There may be multiple other questions that people will try to answer with it, but it's designed to answer typically one question. And for example, for superiority trials, we have a null hypothesis of no difference between arms, and the alternative hypothesis is that there is a difference between intervention arms. So that, for example, in the REVIVE trial, we will expect that azithromycin would be better than placebo. So that's a superiority trial. We've mentioned that sample size requires this we need some certain sample size to have a particular statistical power. So I'm going to be defining what statistical power is, and also talking about what inputs are needed to calculate the necessary sample size. So there are many aspects that may affect the calculation, and there's going to be some statistical aspects. For example, we'll be talking about different RCT study designs giving rise to different ways of calculating the sample size. We also may have multiple hypotheses. If we have a nice simple trial with just one hypothesis, things are relatively easy to calculate. But if we have multiplicity of hypotheses, it becomes a consideration. Also, what is our outcome variable is very important, and how we're going to analyze the data, and whether we're doing multiple tests of the hypothesis along the way. But there's also non-statistical aspects. So non-statistical aspects involve, do people actually follow the or comply with the protocol? Does the staff also adhere or comply with the protocol? We may have subjects sometimes crossing from one arm to the other by not taking the study medication, as they were randomized to. We may have people that drop out. We call them loss to follow-up. And we may have missing outcome data. And these are things that complicate the calculations of sample size. And so there are considerations we need to keep in mind. So there are statistical considerations and there are non-statistical considerations that we have to consider. So let's go back to the basics. Sampling is one of the two fundamental processes of statistical inference. The yellow cloud here, we think about it as the population or somewhere out there in the universe, the unobservable world. And we have characteristics or parameters that we want to describe of that population. But since we cannot observe that population, we have the first statistical process, which is called sampling, which is the process of drawing a sample from that population. And once we get that sample we can observe the characteristics we're interested in knowing in the population. We're going to be able to observe the same characteristics in the sample. And those things are called statistics. And then through the second process of statistics, called statistical inference, we will make some conclusion or some statement about the parameters in the population. You'll notice here that the parameters in the population are Greek letters. And why do we use Greek letters? Simply to confuse people, so they hire statisticians. That's the only reason. But when we calculate the statistics, we tend to use regular alphabet letters. Now, in a randomized trial, what we're interested in is what we're going to call the effect size. And that's in the cloud is delta, little delta. And little delta with the hat is the observed effect size in our sample. So we are interested in what is the effect of the intervention in a population. And we're going to now get a sample from that population and now study and calculate the effect size in the sample, and then be able to go back to the parameters in the population and say something about this is the effect of this intervention in the population. So we have to have sampling and we have to have statistical inference. Now, how do we take the sample is very important. You may have heard that there is probability samples, and probability samples is that a method of selecting a sample where we know the probability of each person being included in the sample. It usually is done in an unbiased way. It's typically representative because it's subject to chance, but if we draw a large number of people, it's likely, and do it in an objective way, we tend to be representative. And there's simple random samples. There's other kinds of random probability samples, proportional to size and so forth. But the point of probability samples that's important is what's here in red. It enables statistical inference. From the sample to the population, now I'm empowered to do the statistical inference. But we also have other types of samples that are called non-probability samples. So usually we draw them from an accessible source, sort of like our patients, for example. But the probability of each person being included is unknown. Many times, as in randomized trials, they volunteer to be, they sign the consent form, they decide if they want to participate, they decide if they don't want to participate, they decide when they see the recruitment efforts that you're doing, they say, oh, maybe that interests me, or maybe not. It tends to be potentially biased, but it's used quite a bit. But the problem with the non-probability samples is that what is in here in red, it's not possible to undergo statistical inference from the sample to the population. Interestingly, non-probability samples is what we do in randomized controlled trials. So then you wonder, why does randomized controlled trials work? The only reason they work is because of the word randomized. Because what we're doing is we're randomizing who is getting what intervention, whether they're getting placebo or they're getting azithromycin as in Revive. So that's what saves the day. That's what enables us to do the inference from a randomized controlled trial. Okay, so that is the basic principles of why we are not taking probability samples in a randomized controlled trial, but we're still able to do statistical inference. The question still becomes is, how do I know the sample size that I need? So when you are going to a statistician for advice on sample size or on anything, there's three places you can go. You can go to them early and you're asking for advice on a study design, and the statistician can provide what we call preventive care advice on study design. It's essentially, as some of you clinicians know, that when a patient comes to you with risk factors but no disease, you can provide primary preventive care. But if you go to the statistician after you've done the study for help with analysis or any aspect of the study, they can only counsel on the analytic tools that are specific to the design that you already have. And it's essentially equivalent to when a patient comes to you with a disease, you can provide some treatment options. The worst case is if you go to the statistician after you're having problems and publishing the results. And so the statistician can only help you, maybe bury the research honorably. It's essentially when a patient comes to you with advanced stage of disease and you say, there's not much we have able to do. We have limited treatment options, maybe palliative care or hospice. So my advice is always come and talk to a statistician early when you're thinking about the study design. And one of the main things in the study design is how many sample do I need in my study? So what aspects do I have to consider when I'm deciding on the necessary sample size? Number one is the statistical hypothesis testing aspects. It'll depend on what type of variable do you have. Are you looking at the binary yes or no occurrence of an event? Are you looking at it in a categorical, discrete one, like mild, moderate, severe pain or level of satisfaction? Are you counting the number of events of something happening? Or are you looking at a change in some biomarker that is measured on a continuous scale? Or are you looking at time to the occurrence of an event, like we're doing in the Revive study? So depending on the type of outcome variable is the type of test statistic that you will use. And therefore, that's one of the main things that drives how we calculate sample size. The other important one is your choice of the outcome and the distributional properties of the outcome, particularly how variable it is. So if the outcome is measured with a lot of variability, like blood pressure, for example, if that was your outcome, your variability would be very large, and we'll see that that impacts the calculation of the sample size. But other distributional properties of the outcome are also very important. Then we have these two possibilities of error in the statistical testing of hypothesis, the type 1 error, which is called alpha. And usually we're only willing to tolerate a 5%, so 0.05. And then the desired power, which is 1 minus the probability of a type 2 error. And that is usually the type 2 error is about tolerable, about 10%, maybe 20%. So the power is about usually 90% that we want, or 80% that we are willing to tolerate, at least have. And we will see, and I'll show you some examples, that the most important driver is what we call the effect size. That's that little delta that was in the upper cloud. You remember? That's the difference between the null and the alternative that you wish to detect. What is the clinically meaningful difference that you expect, that you would like to show your intervention has? The smaller it is, the more impact it will have. So you notice here that we have these 4 Greek letters: sigma, alpha, beta, and delta. And they will come in to what I call the calculations and considerations in sample size. Now, we were assuming we'll have basic study design aspects will also affect things. And so whether we're doing only 2 interventions, like we have in the REVIVE trial, we have azithromycin and placebo, so we only have 2 arms. Some studies may have more than 2 arms, and so the number of intervention arms will have to be taken into consideration. Also, are we doing crossover or factorial designs? Are we doing what type of randomization? And I'll talk about that. And if you have more than 1 primary outcome, those are complications, but let's start off with the simple one of 2 arms and doing that. So here is what I would consider the secret and conceptual formula for hypothesis testing. And I call it, for those of you in Africa, you know what the big 5 are. Well, these are the big 5 for sample size calculation. So it's 4 Greek letters, the same ones we had before. It's the sigma, the alpha, the beta, and the delta. And that will give rise to its sample size. This formula on the left is a specific formula that comes from continuous outcomes, but it also involves, as you see here, I don't know if you're seeing my pointer, you have the alpha here, the beta here, the sigma, and the delta, and here is the n. So the 5 for a specific example of continuous variables, you get this formula. I like to think about it as these 5 elements here. And I like a conceptual formula. Conceptual formula is this. If the only thing you remember from the seminar is this, I'll be happy. And that is, you've got that the necessary sample size is related to the desired power, 1 minus beta, the outcome variability, how much variance there is in that, that's related to the sigma, the desired significance, that's your level of significance alpha, and the difference to detect squared, and that's your delta here. So think about this here. There's 5 things here. The more power you want, since it's in the numerator, the larger your sample size will be. The more noise or variability you have in the outcome, the larger your sample will be because it's in the numerator. The smaller your significance, then the larger your sample size. If you wanted not only a 5% level of significance, but you wanted a 1%, then your sample size will be bigger. But here's the driver. If the difference that you are able to detect, the effect size, is smaller, you better have a very large sample size. And it's also squared. So the impact of the difference is larger than that. Let's look at some examples of this. This comes from the standard error of Y, and the reason we have Z here is because most test statistics are asymptotically Z. So the conceptual formula is involving these 5 things: the level of significance, the power, the differences delta, and the variability sigma squared. Those are conceptually the main considerations for sample size calculation. So how to look at power and sample size. When people talk about sample size, we say, oh, because I want certain power. So if we go back actually to this conceptual formula, you'll notice that we have 5 things here, right? The power, the variability, the difference, the significance, and the sample size. And if we remember our high school, junior high, maybe, mathematics, if I have an equation that has 5 things in it, and you tell me 4 of those, I can tell you the 5th one. That's as simple as it is. So if you give me the power that you want, you can tell me the variability you have, you tell me the difference you want to detect, and you tell me the significance, I can tell you the sample size. No big complication. So what we do is that the question everybody asks me is, what sample size do I need for a desired power to detect a specified clinical meaningful effect? So I could conceptually, I would put them here. The formula, you don't put any numbers in this conceptual formula. There's actual formulas depending on the type of outcome. But conceptually, this is what's happened. You'll be able to, if I tell you these 4 things, I will be able to, using the appropriate formula, get the sample size. But if you tell me, no, my budget is only able to handle 500 people. So my sample size is fixed at 500. What power do I have for a specified effect? So I just take those 5 things and I put the power on one side and bring the variance on the other. And so now that with the given sample size that you have, the desired level of significance, the effect that you want to detect, and the variance, I can tell you what power you will have. Just manipulating that conceptual formula. But if you say, oh, wait a second, my sample size is not only fixed, I still want a particular power, then what is the smallest effect I can detect? Now I just sort of play around with it and I move the effect to one side. And so I have the desired power multiplied by the variance divided by the desired level of significance and sample size. And now because the effect is squared, to get the smallest effect, I need to take the square root of it. So you tell me 4 of those, and I can tell you the 5th one. No big deal. Okay. Now, wanted to point out that the sample size considerations in a clinical trial are usually always based on testing a hypothesis, because that's what we're doing. That's what a clinical trial is designed to do, is to test a hypothesis. However, sometimes some secondary analyses in a clinical trial may involve, oh, I want to know the percent of people that have such and such, or you want to estimate a particular characteristic in the people that are in your trial. So you may want to have a sample size consideration for estimating something. And for estimation, conceptually, now we're looking at having not testing anything. We're just estimating just to know, to describe, to explore. And we talk about presenting that estimate with a confidence interval that is tight around that estimate. So we want a certain width of the confidence interval and a desired confidence, typically 95%. And so the conceptual formula here is slightly different. There is no beta because there is no testing, right? And so now the conceptual formula is different. The more variability there is, the larger the sample size, the desired confidence, if you wanted a 99% confidence, it would be larger. And the narrower, the more precise you want to estimate something, the larger your sample size. But I will not mention this more. I will concentrate on the primary test of a hypothesis, sample size consideration. So now let's go back to, so that was the formula. There's nothing strange. There's nothing fancy about calculating sample size from a basic standpoint. That conceptual formula applies to every calculation. There's not a single one that doesn't use that conceptually. Now, the actual formulas for calculating them are pretty complex. When I was in graduate school, we had to look at 4 volumes of books that had formula for different situations. And nowadays, it's on software packages. We don't have to worry about doing the actual calculation because all the formulas have been programmed by somebody. What we have to understand is what goes into considerations that go into which formula I want to use, and then choosing that formula I want to use. What are the considerations? The design. The basic 2-arm parallel design, just like the REVIVE trial, you take a bunch of people that consent from your target population. The process of randomization gives you the ability to do inference to, in our case, it's placebo and azithromycin, and we follow them to time to development of an event. We're comparing 2 independent groups. This is the most common RCT design, and this is what most standard calculation of sample size can be. Could not be any easier than this. But let's look at some examples of what drives things. So I'm taking now an example for a continuous outcome. Continuous outcome could be like obesity, BMIs, for example, could be blood pressure, change in blood pressure. It could be their satisfaction on a scale from 0 to 100 on their treatment, whatever. But it's a continuous outcome. And so what we're doing is comparing the mean in the intervention arm versus the mean in the control arm. If it's a superiority hypothesis, they're saying no, there's no difference. The alternative is there is a difference. What is a meaningful, clinically meaningful difference you wish to detect? Here we need to give what is the measurement of variability of that continuous outcome, the sigma. We specify alpha, usually 0.05. We specify beta, usually 0.10 or 0.20 or somewhere there, so that the power is either 80 or 90% that you will tolerate or that you would like. So here is a graph where I've given some options, and I have options for different clinically meaningful differences, delta here. The sample size is here. I've said that the alpha is 0.05 and the variance is 5 in this arbitrary example. And I have 3 different powers. What do we notice? What I'd like you to see is that the power, the larger power is the blue, the smallest power is the red. So for the larger power requires a little bit more sample size than the lesser power. But the difference is not that great. The big driver here is that, as I said, the difference between the effect or the effect size, the difference between the arms. For big differences, I don't need big sample sizes. It's easy to detect big differences. But as I go to the left here, the differences get smaller. And as they get smaller, it jumps up. And so when differences are smaller, then the sample size is a lot bigger. Okay? What is the difference that your intervention is going to get? If your intervention is going to big effect, you don't need a big sample size. If your intervention effect is relatively small, then you are going to need a big sample size. Let's look at another example. Most people don't do continuous outcomes in clinical trials. A lot of times is the occurrence of an event, yes or no. So that would be a binary outcome. And we are comparing risks, the risk of occurrence of an event in one arm versus the risk of occurrence of an event in another. So for a primary outcome, we're looking at the proportion of people that get the event in the intervention arm minus the proportion of people that get the event in the control arm. We think that in the intervention arm, if it's something event is good or bad, we will have the difference of them is different from 0. Many people like to think about it in terms of relative risk as opposed to differences in risk. So we just take the ratio of them and we look at relative risk. And now I've done some different examples. You have to specify what is the risk of occurrence of the control arm. So in this case, for example, would be the placebo arm, and the smallest clinically meaningful relative risk you wish to detect. If the effect is very big, the relative risk is going to be very big. If the effect is very small, the relative risk is going to be close to 1. The closer we are to 1 for relative risk, that's the red here, versus the 1.5 is a very big relative risk. The relative risk that's small is 1.1, that's the red. And you notice that for the smallest relative risk, we need relatively more sample size than the other. Okay, so the same thing here. The smaller the effect is the big driver, makes me go up quite a bit dramatically here. Now, in Revive, we have time to an outcome. And time to an outcome is called, time to an event is called a survival outcome. And now what we're going to do is we're comparing hazard functions. So the hazard is what's called the instantaneous risk of something occurring, given that it hasn't occurred earlier. And we use another Greek letter here, lambda, for it. And we tend to not talk about differences in events hazards, but in the hazard rate ratio, and that is the ratio of the hazard in the intervention arm versus the control arm. And again, we will have to specify what the hazard is expected to be in the control arm and the smallest hazard rate ratio that we wish to detect. And we see here that the closer we are to 1, that's a smaller hazard rate ratio, becomes a lot larger sample size. And it does change quite a bit depending on the hazard rate in the control. And in Revive, that's what drove, for example, the need for 8,000, because we're looking at a hazard ratio that is relatively small. And so that and hazard rate ratio that probably is around here, that is going to require around 8,000 people, for example. So for those of you that may not have seen or understand what a hazard rate is, the hazard rate is, if I look at the survival function, the survival functions are usually a non-increasing or sort of decreasing function that theoretically looks smooth like this, that is tapering down. And the hazard is related to the rate of decline of that hazard function. So here the hazard is very steep, and here the hazard is not so steep. And so that's where we get, and we want to know how fast is the event occurring in the azithromycin versus how fast it's occurring in the placebo. And that's where we get the hazard rate ratio. So those 3 examples for continuous, for binary, and for survival outcomes give us an indication of how these 5 things play around with each other, these considerations. But I mentioned that there are other considerations that come into play. One of those is, what type of hypothesis do we have? We typically in clinical trials have superiority hypotheses, but we may have, and it's actually becoming quite common, to have non-inferiority hypotheses. We rarely do equivalence one. Equivalence means that the difference between the intervention and the control arm is less than some value gamma. But it could be actually better, but if it's greater than the gamma, even if it's better, we would not say that it's equivalent. So equivalence trials are usually in the biopharmaceutical area when they're looking at comparing a known drug to a generic drug, for example. But in most clinical applications, we either have superiority hypothesis or non-inferiority. Non-inferiority says, yes, I don't need to show that my intervention is better than control, because the control may be already a standard of care that is well accepted and it's pretty good, but my new intervention is just not inferior to it. It is an alternative that we could use in case it's, it maybe has fewer adverse events. It may be easier to take. It may be cheaper to take, for example. And so I will tolerate a little bit of being maybe a little bit worse, but not too worse. How much not too worse? This capital delta here. And so while in the superiority hypothesis, we have this little delta, which is the smallest clinically meaningful difference that we want to detect. Here in non-inferiority trials, we have capital delta is a maximum tolerable difference that we are willing to tolerate, for example. And usually that difference, capital delta, is quite big, quite small relative to the little delta. So what ends up happening is that for showing superiority, we have little delta, which is a lot bigger than this capital delta, which in turn is greater than the equivalence gamma. And so what that implies, if we remember that the effect size, the smaller it is, the larger the sample size by our conceptual formula. So therefore, when we talk about realizing that non-inferiority hypotheses require a much larger sample size than superiority hypotheses. Equivalence is even higher, but we tend to talk about clinically with non-inferiority versus superiority. So the type of hypothesis is also very important. And that's also part of in the protocol. That's what comes in when we're doing the calculation. Are we using little delta or are we using capital delta? Now, other types of considerations. We've talked so far about the classical two-arm, parallel arms trials, but there are other types of trials. The REVIVE trial is a relatively simple two-arm trial. But in other trials, we may have multiple types of interventions that we want to compare. Maybe intervention A is a drug-based intervention, and intervention B is a behavioral change counseling intervention, for example. And so we want to test them simultaneously and look at the synergistic effects of them. It's called a factorial design, when some people get none of the two, they get the control arms for both, or they get the control arm for A and they get the intervention part of B, or they get the intervention of A and the control arm of B, or they get both interventions. In the presence of no interaction, then we can answer two questions for the price of one. So we gain sample size. We sort of double our sample size. We can do two studies in the same, two separate studies in the same one. But if there is an interaction, now we get no gain on sample size. So many people like the factorial because they can study synergistic effects, or they can also gain on sample size. But if you have an interaction and you have no gain, you basically have a four-arm intervention study. And you could even get a loss if one of those arms requires a lot of people, for example. Now, another interesting type of design that is trying to address the sample size is considered quite attractive, and that's the crossover of subjects. That is, subjects are randomized to A or B, and after a certain period, they have, we call it a washout period. Then the people that got A initially in the first period are given B in the second period. People that were gotten B in the first period get A in the second period. So there's double the number of people that have A's. If I have 100 people, 50-50 here and 50-50 here, now I have 100 people that got A and 100 people that got B. And so I've doubled my sample size. Well, this is possible in some situations, but not in our trial or many trials. Why? Because if the period to observe the event is a long one, now I've sort of doubled the amount of follow. If what I'm looking at is an event that is people, their disease progresses, now they don't go back to their initial state, this doesn't work. If we have people that die, we can't resurrect them and have them do it again. And the effect of whatever intervention I have has to be able to disappear. So while this may sound attractive, it's not usually possible to do in most clinical settings. It depends on the interperson correlation between one period and the next period. So this is, people think about it as a very attractive approach. It's not that usual then. In some interventions, like for example, in hypertension, when you're trying to lower blood pressure and using drugs only, then you have drug A versus drug B. And we know that people, once they're off the medication and they're in the washout period, they go back to having high blood pressure, and then you can do this. So this is done in some types of studies, but not in the typical chronic disease studies that we tend to do that are event-driven, like the REVIVE study. Now, how we randomize people also is a consideration we have to worry about. Simple randomization, where people are assigned with equal probability to each arm, is what most sample size formulas assume. So the actual sample size formula, not the conceptual one applies every time, but the actual formula depends on how we randomize. And most sample size formulas that you find on the internet or whatever assume sample size done by simple randomization. However, if we do stratified randomization, the subjects are first grouped into some groups and then randomized within each stratum, and then increases the uniformity. So we lower the standard errors and we get a gain on sample size. Cluster or group randomization, we have people in clusters that are randomized, and so we lose effectiveness due to the design effect. So stratified, we stratify people, and then some people go to A within the strata and B. And it's very common. This is what we do in multi-center studies. The strata, for example, for us, are the different sites, and we randomize within sites. It gain in power, but we usually conservatively ignore this. Cluster, on the other hand, is when an entire clinic goes all to A, and then another clinic goes all to B, for example. And the big issues are that we lose power here in this case. Why? So the necessary sample size for a regular RCT is this conceptual formula that we had before. But for a clustered RCT, we have to multiply the variance by this thing in red here. And this is called the variance inflation factor, which involves the average number of people per cluster and what's called the intra-cluster correlation coefficient. And this is always greater or equal to 1. And so it becomes something that makes the necessary sample size bigger. So if everything else is unchanged, because we're multiplying this by something that is greater or equal to 1, the necessary sample size will be increased in clustered RCTs. Now, if you don't change anything and you don't want to increase the sample size and you leave it at what it was, then your power will be decreased. How do I know that? Because I just take that conceptual formula and turn it around and put power over here, and we see that because this is here in the denominator and is greater than 1, it's going to make this now reduced. But if you want to maintain the power and maintain the sample size, then the effect that you will be able to detect will be larger if all else is unchanged. Why? Because this will be in the numerator now, and the effect that the smallest effect that I can detect will have to be a bigger one. So again, that conceptual formula helps us understand the difference and the impact of doing randomization by clusters. So I see that the time is running out, and I have a few examples of adjusted sample size in cluster randomized controlled trials. I'll leave the slides with you. You can take a look at that. But basically, it's showing you that we calculate the interclass correlation coefficient, and that the way to do it is you calculate your sample size necessary based on simple random allocation, and then inflated by this design effect or variance inflation factor. And it can be quite dramatic. So even with a small, if you have about average of 25 people per cluster, like for example, in school settings, when you do schools as a clusters, or you do clinics and you get 25 people per clinic, even for a relatively small intercluster correlation of 0.01, you have to inflate your sample size by 24%, for example. And then I have a few examples of actual calculations of how you go determining what is best to do. Do I get more clinics of fewer patients or fewer clinics of more patients? I have to now consider not only those 5 things, the 4 Greek letters, alpha, beta, sigma, and delta, but now I have to consider the intercluster correlation, and don't forget the costs. So this will help you determine if I have a fixed number N that we can budget, how many groups and how many members per group. And then you'll have to worry about attrition and so forth. So I have some few examples here, but I will not go through the examples. I'll leave you the slides and you can look at that. But I wanted to mention that there's other types of ways besides the randomization being the issue. People are now doing what are called adaptive randomized controlled trials. And adaptive ones is when we use accumulating data, decide how to modify aspects of the study without undermining the validity and integrity. So we have different names for this: multi-stage trials, dynamic trials, flexible, response-driven, but requires that we do multiple stages. And then results of one stage guide what happens at the next stage. So the data may be analyzed and the next stages may be redesigned, taking into account the available data. And so people do this adaptation by design. And one of the things they like to adapt is sample size. And so we do a lot of, during the recruitment stage, they may adapt the eligibility, the allocation, but they can adapt the sample size based on what they've observed so far. Do I still need to recruit as many as I need to? During the conduct stage, after you've already completed the sample size that you have, you can maybe adapt the sample size. No, you've already recruited them, so you cannot adapt the sample size. What we do adapt is when we end the trial, when to stop the trial. This is the interim analysis that is usually done, okay? We may sometimes during the study not usually decrease the sample size because we already had it. We may be able to increase the sample size and we adapt it there. So the impact on sample size is we may adapt allocation or changing the eligibility, may increase the sample size, or interim analysis are adapted at the end of the trial. And so doing those interim analyses that we are usually forced to do, we need to, so if we do like 5 looks, we may have to increase the sample size by 1.5% or maybe even 3% if we're doing about 10 looks to the trial. And so it's not that much, but it's somewhat that we have to do. So finally, other adjustments to sample size calculations is for expected incomplete data, for expected people not following the protocol, for specified multiple testing, and for exploratory analysis. So like it's in subgroups, etc. Multiple things that go beyond the standard one single question that a clinical trial is designed to. But this is what makes our job interesting or complex. This is why you come to a statistician early. Other final considerations: if you have complicated designs of more than 2 arms, if you have a factorial design, if you have more than one primary outcome, if you have to impute outcome for too many participants, and the US FDA has how to deal with multiple primary outcomes and secondary outcomes. So I will finish that and say, thank you very much. Bye, Duncan. Sorry I've ran out of time, but I still can entertain questions if we have time.
Thank you, Professor Bangdiwala, for your time and this insightful talk. I see Dr. Sean Wasserman has got a question.
Thanks, Elsa. Hi, Shrikant. It's Sean. I'm speaking to you from Entebbe, where we're doing a site visit for Revive. Hello to everyone. That was a great talk. Thanks so much. I have a question about factorial designs, empowering them. So you've spoken about, you make an assumption that there's no interaction, and you power the trial for one intervention. Does that mean that you have to make an assumption about the effect size of the factorial of the other intervention that you are randomizing? So that has to be the same if all other things are equal, right?
Right. And so actually, usually they're not the same, right? So you first of all assume no interaction, and you say, what is the effect that intervention modality A will give me? I power that, and maybe I need 400 people per arm. But now I want to also look at intervention B, and the power for that one may be 600 per arm. So I better do the 1,200 people, and so I may get different power than the other one. So you power them separately and take the largest one of those, assuming no interaction. If you have interaction, then you'll have less power anyway. But if you're interested in the interaction, now you power for the interaction one, and there's ways to power for the interaction one.
Sure. So the bottom line is that trial is powered for the intervention with a smaller effect size.
Correct.
Okay, thank you.
We have got time for one more question if there is any more. And if not, I just want to repeat that you can either pop your email in the chat box, or you can email me. My email address is indicated there. I can then share the recording, or Professor Bangdiwala also agreed that we can share the slide set with you. So please either get in contact, and then we will make sure that these reach you soonest. I think I don't see any other questions. Thank you everyone for joining today. Yeah, and please reach out if there be anything that we can assist with.
Okay.
Thank you.
Thank you
Have a
very
good
much.
day.
Bye-bye.
Bye.